Good morning, everyone. My name is Dominic Rame, and today I'm going to be presenting primarily on Legionnaire's disease, with a mention also for its less severe cousin, Pontiac fever. So the taxonomy of Legionella here, it is in the kingdom Bacteria, family Legionellaceae, genus Legionella, and species Legionella pneumophila. Pneumophila is the one that is primarily the cause of Legionnaire's disease, and there are other subspecies of it, but for the purposes of brevity in this presentation, I will be only looking at Legionella pneumophila. So as before, it is a bacteria. There are no colloquial names for it, no nicknames or shorthand. As far as I know, all I could find on it was that it is Legionnaire's disease. The symptoms and signs are those of acute pneumonia. This can make it very difficult to diagnose, and it often goes misdiagnosed or undiagnosed. Further characteristics include that the growth factor for this is L-cysteine, and also ferric iron is key in its proliferation. It metabolizes amino acids as opposed to carbohydrates. These are some images of the bacillus itself. You can see there's a single polar flagella here, and then this is an actual gram staining of it. As you can see from the light pink, it is a gram-negative bacteria. Continued characteristics: gram-negative bacillus measuring between 2 and 20 micrometers in diameter. It is motile by means of a single polar flagella, as on the previous slide. You can see it there on the end. And it has additional motility provided by fimbriae. It is found exclusively in freshwater sources and is unique in the sense that it can proliferate in the presence of phagocytic cells such as macrophages or monocytes. As to the notable strains, there are more than 58 of them. 25 of them are known to cause illness in humans. The serogroup specific that cause the most severe illnesses in humans are 1, 3, 4, and 6, with 1 being the most severe. Pontiac fever is caused by L. felii and L. longbeachii, and that has symptoms and signs similar to the common flu. All species can be inhaled via water spray, so aerosolized droplets. And as far as I could find, there are no instances of it being transmitted from person to person. The sources that I did find said that it is possible under very rare circumstances, but did not elaborate any further than that. The pathogenesis, it starts with a contaminated water source. Inhalation of infected droplets are brought into the lungs, into the alveolar spaces, where they're picked up by alveolar macrophages, which engulf them into phagosomal vacuoles. By an unknown mechanism, L. pneumophila prevents the fusion of a lysosome to the phagosome, which prevents the destruction of the bacteria itself, and this becomes a key component to its reproductive cycle. Once it's neutralized the lysosome and the macrophage itself, it proliferates within the phagosome. So it uses the macrophage's own internal mechanisms to basically protect itself from destruction, and it proliferates inside the macrophage itself until it reaches a point where it can burst out of the macrophage and infect further cells. And these are some of the images here of the life cycle. Here you can see it being engulfed by the macrophage. It's secluded in a phagosome. You can see the lysosomes trying to bind to it, but it basically uses the lysosomes as a barrier. It prevents them from actually fusing with the phagosome properly, which prevents them from acidifying the internal contents of the phagosome itself. And then here is an actual microscope. It's an image from a microscope of what an infected cell looks like. What the cell looks like inside. The incidence and prevalence of it. The United States records roughly 10,000 cases per year. I specified here 2018, but that's generally consistent from year to year. It is suspected that the case count is actually about 1.8 to 2.2 times higher than the reported case count because of undiagnosed or misdiagnosed cases. The European Center for Disease Control reports approximately 2.2 cases per 100,000 population, which is an increase from 1.4 cases per 100,000 population from the year 2015 to 2019. Spain, France, Germany, and Italy accounted for about 71% of the cases, which is I found to be particularly surprising because those countries are all developed countries with access to clean water and proper purification protocols. I actually expected to see less developed countries in that particular spot, but for one reason or another, they're not there. Legionella was first discovered by Dr. Joseph McDade in December of 1976, which is the same year when the initial outbreak of Legionnaire's disease occurred. In July, there was an outbreak at a convention center for the American Legion at the Bellevue Stratford Hotel in Philadelphia. This is where it gets the name Legionnaire's disease from. There were a total of 221 cases and 34 recorded deaths that resulted. The hotel that was used as the venue was actually closed as a result of the investigation into the outbreak itself. As far as the most recent outbreaks that I could find, there was a recent case in New York City, or recent cases in New York City and Harlem, where an infected there were area cooling towers that contained contaminated water samples. And this was on September 22nd of this year, the cases were resolved. With 18 total cases, 16 of whom were discharged. There were no recorded deaths from this outbreak. A little bit further into outbreaks, what constitutes an outbreak is 2 or more people exposed to Legionella in the same place and getting sick at about the same time. They generally occur in or around complex structures structures or buildings with complex water systems such as hotels, resorts, long-term care facilities, and hospitals, which indicates to me that this is often a nosocomial infection resulting from improperly sterilized equipment, such as a nebulizer or a respiratory aid device. Once more, it is transmitted via water droplets small enough to be inhaled. Rarely, what can happen is sometimes people who are drinking water, they may inhale water into their airway as opposed to down their throat, and it can get into their lungs. The body does have a natural defense against this. Your lungs try to force you to cough those particles back up, but people who might have swallowing-related difficulties are going to be at a greater risk because their body might not perform this function to the maximum effectiveness. Once more, only under very rare circumstances is it transmitted from person to person. I could not find any particular cases or reports about it being transmitted from one person to another, but I can speculate that it would involve transfer of fluids from one person to another, likely orally, so saliva, essentially. The most at-risk populations for this, as with most cases of pneumonia, is going to be elderly populations or perhaps very young populations, although in that case, the latter case is less likely. The age range, however, of 65 and up is by no means exclusive to this disease. It can and does occur in at the same level of severity in younger ages, 20s, 30s, 40s, 50s, and likely even in teens. Developing nations or nations without access to clean water are going to be particularly susceptible, considering that vigilant monitoring of water purification systems is essential to preventing an outbreak. Currently, there is no vaccine yet available. There have been tests run with guinea pigs. In vivo experiments have been run on guinea pigs, and there have been positive results yielded from those experiments, but there have been no clinical trials conducted as of yet. Eliminating it at the source, meaning the water source where it's found, is generally going to be the most effective means of control. But it can be costly in terms of resources and risks. The most effective means of preventing it is vigilant monitoring of water supplies and water purification systems, in addition to other standard methods of pathogen prevention, such as hand washing and filtration. Standard defenses such as alveolar macrophages and recruited blood monocytes are not going to be as effective in their general role as they otherwise might be with any other pathogen because of the role that they play in the proliferation of the disease. One of the things that I found interesting about this is that it requires ferric iron to reproduce. So antibiotics or treatment methods that inhibit the passage or implementation of ferric iron into alveolar spaces they are an effective means of controlling this pathogen. Strong specific and non-specific responses are also very important in an immune response in Legionnaire's disease. Scientists and researchers have not yet concluded what component to the immune system is essential in preventing the spread of Legionella infections, but it is strongly suspected that cell-mediated immunity is the direction that they ought to be looking, specifically because of the pathogen's nature as a facultative intracellular parasite. Humoral response is a bit less clear. Antibodies have been studied both in vitro and in infected individuals. They don't appear to have the kind of effectiveness that they might otherwise have against any other sort of pathogen. And in some cases, they can actually cause more harm than they can good. The complement system is also more of a risk than it is a benefit because of the role that it plays in exacerbating the growth of the pathogen. It accelerates its proliferation immeasurably. The symptoms of Legionnaire's disease are going to be mostly the same in most cases as pneumonia, and will include cough, muscle ache, headache, fever, shortness of breath, and chest pains. They begin 2 to 14 days post-infection, but can take more or less time depending on the case. Convalescence or recovery from the disease itself can take anywhere from a few weeks to several months of antimicrobial therapy, and it depends on the health of the individual in question. It depends on their age, their predisposition. It depends on any number of factors. It can be, and most often is, detected via chest x-rays, the same way that you would detect pneumonia ordinarily. There are generally 2 tests that are run in tandem with chest x-rays, those being the urine tests and sputum tests. Lab cultures are going to be the most reliable methods of detecting, but require specialized media and equipment to be prepared ahead of time. Morphologically distinct colonies can be presumably identified as Legionella if they rely on cysteine for growth. The treating of Legionnaire's disease is a tricky business because there's no vaccine and because of the fact that it requires oral antibiotics at the moment. Oral antibiotic therapy is the only way to really treat Legionnaire's disease. Those that are most effective include fluoroquinolones such as levofloxacin, moxifloxacin, macrolides such as azithromycin and clarithromycin, and tetracyclines such as doxycycline. Initially, erythromycin was used to treat this disease, but because of the health risks involved in prolonged exposure to that particular antibiotic, it is no longer the primary source of treatment. Continuing research efforts, as stated before, there's no vaccine, and there has been research done towards a vaccine for quite some time. The specific journal that I found cited 1984, and this one, like most of them, uses guinea pigs for in vivo protection experiments. Researchers are continuing to work towards determining what components of the immune system is specific to an immune response, because at this point in time, we don't know exactly what it is. As said before, cell-mediated immunity is suspected to be the key. And the other thing that they're looking towards is what mechanism exactly allows L. pneumophylla to prevent it from being destroyed by a macrophage-bound lysosome. And these are just my references. So I hope that you enjoyed the presentation. I very much enjoyed giving it. I hope it was informative. Thank you very much for listening, and have a wonderful rest of your day.